Um, this is going to be a fairly informal talk, so please feel free to ask questions. I'm going to use the board. This is something that I did a few years ago with Michael Desai, who's here in the room. I'm going to explain what I mean by those terms eventually. In the spirit of the conference, the data is going to be big, but the models are going to be really simple. They're all linear and Gaussian, but the data is big. All right, so the central question that we're interested in is to ask, is evolution repeatable, right? So the key question is evolution repeatable? Right, so if you kind of went back in time, maybe 1 million years, there's some time scale, you define a time scale, and you start doing everything that you would do again, would you see the same things, right? So of course we can't really do that because we can't go back in time, but we can do experiments in the lab, right? So we can set up lab experiments where we can ask this question in very controlled conditions, and that's what folk have done. And in a typical lab experiment, in experimental evolution setup, the way you do it is that you could take a collection of cells and you put them in a flask. This flask has certain nutrients, right, that you've defined. You let them grow. Typically, they grow exponentially before they stop growing because they run out of nutrients. And after a while, you transfer them to a new environment, maybe a day later. And you do this over and over again. Yeah. So this is a serial dilution experiment. And typically, this can be 1 day, 2 days. Right? Let's say it's 1 day. You can do this experiment for many days. You can do this experiment for many weeks. And folks have now done experiments for 35 years, right? So that's the Lenski experiment. And so there's a lot of data coming from these kinds of experiments. Okay, so if we go back to the question about repeatability, what are we asking, right? So we're asking something about some notion of universality, because we have to define what is repeated, right? So of course, certain things are not going to be repeated, but so we need to work with the right level of abstraction. So the question is, what is that? You can ask that question at different levels. So you can ask this question at the level of behavior or phenotype. These are coarse grain observables that you can measure in experiments. You can ask this question at the level of circuits, algorithms, or pathways, some intermediate layer of abstraction, right, that I'm not going to really define here. And then you can think about repeatability at the level of parameters, so to speak, or genes or mechanisms through which collections of genes operate, right? So there's different levels. It's not a discrete set. There's a continuum here, but you can ask at what level is evolution repeatable, right? In the context of these experiments, let's say we want to look at behavior and phenotype. So the question is, what is a relevant phenotype to measure? And the classic thing you can do is to measure some notion of fitness. And when I say fitness, these cells on average, they grow exponentially. So if you take a collection of N0 cells and measure the number of cells sometime t later, this is going to look something like this. So this is essentially the instantaneous rate that these cells grow. So this is the instantaneous growth rate. If you have a population, you can take the average. And so you can always write your final numbers in this form. And so you define some notion of an average growth rate. where now your capital T is over very long time scales, right? So you kind of want to average over any fluctuations in the environment that you can have. So in the context of this experiment, you want to average over many days, right? Because in each day, you're going through a set of nutrient conditions that these populations experience. So that's the phenotype that we're going to measure. So this is what we call fitness. The idea being that in the long term, the cells that have the largest fitness are the ones that you're most likely to see when you sample from a flask a long time later, right? So it's an empirical definition. All right, so folks have done this in different kinds of experiments. I'm going to show you what the fitness trajectories look like for the Lenski experiment. So the Lenski experiment was done in E. coli. This is a bacterium that you can find in your gut. And this experiment was done it's still ongoing. It's been, I think, 36 years now. And I think they're at about 75,000 generations, if I'm right, but the measurements were taken at 50,000 about 10 years ago. And in this case, there were few different populations of E. coli. So it's a clonal population in the sense that all cells have the same sequence, right? So when you first put in, but eventually they acquire mutations and some of those mutations get selected. So as time progresses, you're going to get a very diverse population of cells. So if you look at the average fitness relative to the ancestor, so this is going to be 0 at time point 0, you're going to get some curve that looks like this for one of these experiments. Now, if you measure this for one more population, it turns out that you often see something very similar, right? So and they've done this for, I believe, 8 different populations, and often you see quite convergent trajectories, at least at the fitness level. Yeah.
Just a technical question. It's true for the mutator lines too?
Yes. Yeah, that's an interesting question, but there is one line that is quite interesting. So at some point, it kind of figured out how to metabolize a nutrient in the medium. And that one, if you look at it, it goes like this and then it shoots up all the way up. So there's one exception, but the majority of them follow reproducible trajectories.
I have a question.
Yeah.
You may be coming to this, but you've sort of implied that you're talking about a fitness at an individual cell level almost.
Yeah.
But in fact, you can think about a fitness in a population where you have diverse cells that are doing different things.
Yeah.
That I think is very biologically interesting.
Yeah.
And then the exponential growth it's more complex, right?
Yeah, I would say that this average over here is hiding a lot of things. It's hiding an average over the population, but it's also hiding an average over environmental fluctuations and a lot of things, the interactions between the cells in that culture, which I'm hiding under the rug, at least in the context of this experiment. Yeah.
Do they ever try changing the medium, the balance of things?
Yeah, so Michael has done a bunch of these experiments. Others have done them too. It was not done over here. In this one, there's one particular medium that started 36 years ago, and they kind of kept at it, right? And someone has to do this by hand, by the way. So someone has to go into the lab and make sure the transfer happens. Okay, so this is an E. coli. I'm going to describe briefly something that Michael has done maybe 10 years ago. This is in yeast. It's slightly different. It's much larger in scale in terms of the number of populations that you can measure, but much shorter in the time window of evolution. It's 250 to 500 generations. So what Michael did was to generate strains that have different initial fitness. Okay. And run this experiment multiple times for each of these. There's some complexities here that I'm going to ignore. So what you can measure is the gain after 250 generations, let's say. And what you see is something like this. So this is after 250 generations. And they made one more measurement after 500 generations, and you see something like this. Okay. And there's two things that are interesting here. One of them is that the gain in fitness. So one thing you should notice is that each bin here has multiple different genotypes, different strains. They're only conditioned based on their initial fitness, but they all end up at the same place, roughly speaking. There's error bars here, but they roughly end up at the same place. And there's also this negative trend over here, which is that you can predict where they will end up based on their initial fitness, right? So it's quite, in some sense, simplified at the fitness level. But of course, if you sequence these populations, there's a lot of different things going on, right? So in both of these cases, it turns out that at least when you measure fitness, this is repeatable. And now we have sequencing for this, at least at the bulk level, and more precise sequencing for this, so you can identify where the mutations happened. And it turns out that at this level, it's not very reproducible. Okay, so there's something happening in between that is somehow averaging out the fluctuations over here and giving rise to a reproducible phenotype, which is what we want to figure out. So there's a question. Yep.
I was curious, I don't want to take you on trust, but
Yeah.
that plot of initial gamma versus gaining gamma, the way you draw it looks like it hits zero. Do you think that's true? Do you think there's kind of a maximal gamma that you would eventually hit and you would get no further gains?
Yeah, presumably there's at some point where you kind of, you still get smaller and smaller amounts because you're always selecting for, mutations that have an effect. And so they might have really small effects. But if you go to a very large fitness, it's possible that you kind of reach a dynamic balance where the number of deleterious mutations that you generate are so many that they balance out the few beneficial ones that you have. And folks have shown that this is some kind of ratchet that happens. You kind of reach a dynamic steady state. So you can end up at zero, but you're going to see fluctuations around here. Right. Yeah.
I have a question about this diagram.
Yeah.
So if you take one of the point on the x-axis at the beginning, and then you do 250 generation, after 250 generation, do you have more than one genotype?
Yeah,
Yes.
you have many.
So when you say the gain, is it the gain of the best new genotype or the gain of all of them?
Either sample from the population, you create clones from them, and then you measure their fitness. And here there's a few averages taken. There's multiple averages being done over here. There's different strains in the beginning set. Once you get here, and for each set, there's multiple experiments. And from each experiment, you get multiple clones. So there's a few averages being done here. Yeah. Okay, so the key point I want to make over here is that there seems to be reproducibility here, but not necessarily here, right? Okay. So what's going on in these experiments? There's a lot of things going on, right? So these curves look nice and clean, but if you think about at the molecular level, at its core you have mutations. These are random, right? So whenever you're sampling mutations on your genome, for yeast, it's about 12 million base pairs. So there's many different possibilities. So one way to think about mutations is to think about the distribution of fitness effects, right? Because that's all that matters. So you're sampling your delta gammas, the fitness gain that you will get from that mutation, from some distribution, which depends on your sequence, your existing sequence, right? So this is called distribution of fitness effects, or DFE, and this depends on the genotype-phenotype map. So this is really a function of how the organism is organized, right? Okay, so now once a mutation is sampled, it appears in your population. There's various forces of selection that favor that mutation if it has a beneficial effect. So this is a quite complicated process when you have many different strains in your population. You can have all of them compete with each other, and in some cases they interact. And you can have many different mutations kind of segregating at the same time. So this process is quite complicated, but I won't address this in this talk. Okay. So we're going to focus primarily on the genotype-phenotype map and the distribution of fitness effects.
So you assume this map is also not dependent on any kind of frequency and
Yeah, we're not going to assume frequency. Well, we're going to make measurements of just this. The frequency dependence comes over here.
this map would also theoretically be dependent on this, right? You can't just assume it's a stationary map.
Yes, so here the fitness would be the fitness of you take a single sequence, you mutate it, and you compare its fitness against that one ancestor. Yeah. Of course, they can interact too, but we're not going to consider
The
that.
fitness depends on the other cells.
Yeah, so I think that's related to the question about frequency-dependent selection. That could happen. And in the experiments, there's a lot of care taken so that it does not happen, or you kind of quantify to what extent it happens. And in many of these experiments, the frequency dependence is quite minimal. Right, so the interactions between the different strains is minimal, but it can happen for sure. Okay. All right. So here I wrote the distribution of fitness effects is a function of the sequence. So it means that it's background dependent, and that dependence on the background is called epistasis. It's really just jargon in the field, which just says that there's some interaction with the background, right? So the effect of the mutation is not uniform across all backgrounds, but it depends on the background. So that's called epistasis. That's just a generic term for that. Now, I mentioned global epistasis here. The hypothesis that we want to test is that whether this has certain simplified structure, right? So we want to ask, if you take this distribution, can we write that as P of delta gamma depends only on the current fitness, depends on the sequence only through the current fitness of the organism, right? So somehow the sequence of the organism influences the fitness, and all the changes just depend on the current fitness. Now, if this were true, of course, you can explain both of these, right? So here we're saying everything that you sample just depends on the initial fitness. So if you run this process for a bit, you're going to get something like this. And of course, the delta gammas on average are going to be smaller for larger gammas. So you're going to get some curve that looks like this, right? So the question is this true? Right? And this is in some sense saying something about the global architecture of the cell. It's really an empirical question. A priori, you shouldn't expect any such feature. So we call it global epistasis. Okay. And again, this is really a feature of the genotype-phenotype map, independent of how selection happens. Okay. So now I'll get into the experiments that were done in order to measure this, right? And I just want to also kind of explain a little bit about the different kinds of experiments you can do in this field, because I assume that it's a diverse crowd. So I kind of want to give more of a big picture as well as a more specific question. So let's say you want to measure something about genotypes and phenotypes. And you kind of want to identify some structure in these maps. There's two different things that you can do, broadly speaking. So one thing you can do is that you define a set of what they call loci. These are genomic locations or mutations that you can apply or perturbations. So you define your space of perturbations. So everything that you're doing is operating on the space of perturbations that you can do, right? So that's how we think about this. So you define, let's say, L loci. I'm going to use this term loci because that's what's common in the field, but you can think of them as the different ways you can perturb the system. What you can do is that you can measure different phenotypes. So here this was fitness in a single environment. You can potentially measure phenotype in multiple environments. And so you perturb one locus at a time. So maybe you apply a mutation at this genomic location and you see its effect on multiple phenotypes, something that you can measure. Now, if you do this for many different loci independently, you get their effects on a collection of phenotypes. This is going to be a matrix, and you can do relatively simple matrix factorization approaches to kind of identify low-dimensional structure in this kind of data. And this seems to work reasonably well for how simple it is. So that's one approach that you can take, and the idea is that loci that have similar effects are probably clustered together, right? So this is what this is something that exploits something the folks in the field call pleiotropy, which is this idea that each locus has effects on multiple phenotypes. The other approach that you can take is to use epistasis. And in this case, it's a bit different. So you have a single phenotype, and this is what I'm going to focus on. So I'm not going to talk about that. We've done a bunch of stuff related to this, and we're still continuing to do this kind of analysis of experiments and data analysis, but I'm going to talk about epistasis. And in this case, what you have is that you again have your collection of perturbations that you can do. You have your L loci. And what you do now is that you perturb multiple things at the same time, right? So the idea is simple. The idea is that you perturb this, you measure the effect on the phenotype. Let's say you perturb this, you measure the effect on the phenotype, and you perturb both of them, and you ask how much do these two interact, right? So again, it's acting in the space of perturbations, and you're asking how much do the perturbations interact. And hopefully, if you do enough of these, you get some view of how the cell is organized, okay? So one way to think about epistasis, or one way to represent it mathematically, is using this idea of a fitness landscape. In a fitness landscape, it's defined by how you're doing the experiments fundamentally, right? So you have to define your space of perturbations. And I'm going to call my sequence as X, right? This is the state of the perturbations that you're doing. And you have L sites. And these are Boolean variables, right? So each xi is a Boolean variable. I'm going to, for various reasons, I'm going to say that they're plus or minus 1 rather than 0 or 1. And the way you represent your fitness landscape is this way. And there's different ways you can represent it. So you can expand to all orders. You're going to get 2 to the L terms because there's 2 to the L different possible perturbations that you can do. And when we say we want to identify the structure of this map, what we're really asking is about the structure of these coefficients F. Right? So what we want to do with these experiments, what we want to get from the data analysis is to say something about the statistical structure of these coefficients. And why are these coefficients important? Because they capture some notions of effects and interactions. So this one is really just the average fitness when you average over all perturbations, if you sample them uniformly, all 2 to the L. Fi is the average additive effect of perturbing one locus averaged out over everything else. This one is the average second order epistatic effect, which measures the interaction between 2 loci, again, averaged over all the backgrounds. So these coefficients have interpretations and you can measure them, right? So you can perturb your system in different ways, and you can do your linear regression or whatever you want to call it, whatever you want to do, and you can fit these coefficients. So every system is going to be different, and the goal is not to fit every coefficient precisely. That's really not what we're trying to do here. The goal is to identify certain common statistical structure that exists across many of the systems that we measure. right? So it's more of a meta goal. We want to work with different systems, do these measurements, but ultimately we want to identify if there's certain common patterns across systems, right? So that's really what we want to do. Again, there's different ways one can do this, and going forward, I'll focus on one particular way of collecting data. But just to give you a few examples of how one can, what one can do with this. So for example, what you can do is that you pick a relatively small set of perturbations, maybe 10 of them, so 10 sites on the genome, and you decide to perturb them in all 2 to the 10 possible ways. So that gives you a complete fitness landscape, so you can measure all the Fs, but it's complete, but it's quite narrow, right? So you're getting a very local view of the landscape. But that's something that folks have done. It becomes complicated once you scale up the number of loci, but we're getting better and better at it, but that's something that you can do. One other, I think a little bit more interesting thing that you can do is to break something. So let's say you're interested in how a pathway functions. You break it, you take the most important element out of it, just enough that it doesn't kill the cell, but it's severely impaired. And then you run an experimental evolution experiment, and then you ask, there's certain mutations that are going to arise that fix that pathway. And so you can ask what those mutations are and how much they interact with the original gene that you perturb. And that you can do by making, again, these kinds of perturbations. So there's different interesting ways of doing this. And I find it particularly interesting because it's really, causal effects analysis on steroids, right? So we have many different ways you can do things, and the question is, what can you say what can you say about the system from these perturbations? So, with the goal of getting to here, Michael designed with his graduate student Milo a very particular experiment, which really tries to get at this one directly. So that's what I'm going to focus on. If there are any questions, I can take them.
I have a very naive question.
Yes.
Biologist about the ...
Yes.
That expansion of gamma of X. So in practice, do you think that does biology allow for some arbitrary number of multi-way interactions between the phenotypes? In other words, do you think that what's hiding in the ... is very high degree, or do you think that these low degree terms actually dominate?
I think that's one of the key questions in this field. How much of the ... should we care about? And I think it's very hard to get there because in some sense, each coefficient, there's many of them as you go ... but each one can be quite small.
But it's after the interactions as constant sign. They're not small, so those are not lower order terms, right? They're
The Fs are small.
Oh, the Fs are small.
Yeah, they increase combinatorially, but each one can be small. So if you're trying to fit a landscape, any measurement noise that you can have, so this is a Fourier representation. So any noise is adding basically a constant to everything, and so you have a fundamental limitation on what you can measure. Yeah.
You've mentioned metabolism a few times. You have to metabolize nutrients and get all the way to a new copy of the cell. Usually there's metabolic networks that have a stoichiometry for some step and a regulatory part. And flux balance analysis looks at just the stoichiometry part. Can you sort of optimize that network and use that to explain the fitness function?
That's an excellent question, I think. I think you can do it to some extent. I don't know if, Pankaj, maybe you have a better sense of, yeah, so there's people working on that about introducing enzymatic action and regulatory mechanisms into flux balance analysis.
More like long-term evolution.
Yeah. Well, maybe I'll touch upon that later in terms of why. So there's a bigger question about why we should expect such a structure, a low-dimensional structure. That is a different conceptual question. This is more about inference. How do you get that from data? But maybe I'll get there if I do have time. Okay. So Michael and Milo designed this experiment. It's an elegant experiment in the sense that it uses what yeast naturally do. So if you take yeast, you can take 2 strains of yeast, so orange and yellow. So this is, let's say, some strain of yeast that they use in the lab. And this is a strain of yeast that they use for making wine. And these two are quite different. I mean, they're mostly the same, but they differ at about 40,000 sites. So there's 40,000 different mutations that on this one that's not here. Right? So there's 40,000 SNPs, as they call them, single nucleotide polymorphisms. So what you do is that you mate them. So yeast mate. And when you made them, you get a diploid and you sporulate them. You end up with a haploid, and that haploid has a mosaic of the 2 parents, right? So it's going to have a genome, and one of the progeny can have a genome that looks like this. And you can have other ones that and so on, right? So when you do this experiment, you get a collection of progeny, and each of them has a genome that looks like this. The reason I drew blocks is because they have some correlations. So you really get blocks of linked loci. But you've generated basically a variety of different perturbations, and it's combinatorial, right? So you have your plus and minus ones, but you've created an ensemble where the plus or minus ones are on average uniformly distributed, right, over the 2 to the L possibilities. And those are your backgrounds on which you can perturb stuff. So what they did was, again, to use something something clever. They took this method that indiscriminately introduces mutations into a sequence. So these are insertion mutations. So you kind of insert a base pair in the genome all over the place. Typically, there's just one of them per cell. But what these insertions do is that if they fall in the middle of a gene, they break how the gene is transcribed, and so the gene doesn't work anymore. So it effectively deletes a gene, right? So the perturbations that they're doing are indiscriminate gene deletions. So you can take each of these and you can delete a bunch of genes on each of these backgrounds. So if you have, and these are the numbers from their experiment. So if you have 150 of these backgrounds and you apply these gene deletions, there's about 1,000 of them that you can do that don't kill the cell. You get a data set which is roughly of this size, right? So 150, and for each of them, you have these 1,000 perturbations. And it turns out that in practice, many of these 1,000 perturbations that you do, they don't really have a large effect on fitness. Because you're measuring fitness in a very particular environment. So it's quite likely that the deletion that you're doing does not affect anything at all. And if you filter this out, you can reduce this down to about 100. I mean, 91 to be precise, but it doesn't really matter, right? So you have 100 different significant perturbations that matter. Okay, so what do we get from this kind of data? So the way we plot them is to look at the effect of each of these 100. So I'm just showing one of these 100. So let's call this mutation 1 against the fitness of these 150 different backgrounds. Okay? So because they have diverse genotypes, they're going to have diverse fitnesses. So there's going to be a whole range of background fitnesses. Sorry, this should be called that. And now we plot them like this. So there's mutations that show, basically, many of them show 0 effect, so that we've filtered them out. So you're only seeing the ones that have non-zero effect. So you have ones that have, there's a lot more noise than what I'm drawing, but you get the point. You have something like this. You have mutations that show something like this. And then you have mutations that show something like this. It's a pretty clear linear trend. Yeah. Okay. So what does this tell us, right? So that's the question. And the reason we plotted them this way is because we want to see if there's any patterns in this distribution, right? So that's the reason we're plotting them this way. Now, if you look at this one, what is this saying? This is saying that the fitness effect doesn't depend on your background at all. It has a constant effect. So presumably what this mutation is doing is that it's not interacting with anything that is varying in the background, but it does have an effect. So you're seeing a constant offset independent of what's happening. Now, this one is slightly more interesting, right? So this is like a two-state system. What's happening here, presumably, is that there's one of these background variations that has a huge effect. So depending on whether that's there or not, you're either here or here. And your mutation that you're doing is interacting with that particular locus. And so depending on where you are, you're going to have a two-state effect. Now, this one was a puzzle for us. Okay, so this kind of told us that there's some kind of nice negative linear trend. It kind of looked like regression to the mean, but you know what's really happening here? Why? And we see quite a lot of these, right? So half of the 100 that we have here shows something like this, right? So there's different names that have been given to this phenomenon. It has been seen in different systems, but we wanted to explain where this came from. And we wrote down a model that is quite simple, but it gives us some insight into the structure of those landscapes, which I think is interesting. Okay, so if you think about what's happening here, let's say you have L+1 loci, so your X vector is that. Your X0 is the mutation that you're applying. So you're flipping it from -1 to +1 against a bunch of backgrounds that have ±1 sampled randomly over here, right? So 1 to L, X1 to XL are ±1s with equal probability and uniformly independently across the loci, but the X0 is the perturbation that you're doing. So you can write down your background fitness as just using that. I'm going to write it in a particular form that helps us see what's happening. Okay. It's a very particular form, but it's just a rewrite of what's there. I've introduced a 0th locus. That's the one that we're perturbing. I'm going to call this phi of X without 0, because that depends on everything except the focal mutation that you're applying on all of these backgrounds. And I'm going to call this psi of X given 0. So the argument is really simple. So what are we measuring here? We're measuring delta. So if you rewrite this, I think it's simpler to see it when it's rewritten. So you have phi of X0 plus X0 psi 0. And delta F is simply, so X0 goes from -1 to +1. So you get a 2F0 plus 2. Yeah. Okay, so sorry. So the assertion is the hypothesis that you're proposing is quite simple. If you look at this one over here, it has 2 to the L terms, right? And if it were true that many of these dots were actually non-zero, you're averaging over many, many terms. So we're going to use a very simple concentration argument to say that this is a Gaussian random variable. Okay, so the first assertion is that phi is Gaussian, right? And that's kind of reasonable because you have many, many loci. The second assertion is that psi is also Gaussian. And this is slightly non-trivial, slightly in the empirical sense, like there's no reason why you should expect that. What this is saying is that the focal locus that you're interested in interacts with many of the other loci in the background. Okay, so that's, again, you're applying a concentration argument over here to say that psi is Gaussian. Now, this already tells you what's happening, right? So you have 2 Gaussians, so the conditional expectation of delta gamma given gamma is going to be linear. Now we're going to make a third assertion, which is that the correlations between this and this are negligible. Okay? And this is something we can measure in the data, by the way. So we can just measure this and we see that it's small, right? So we're going to say that when you take your data set and you take these averages, they're negligible. Right, so there's just a correlation coefficient. Now, if all of this were true, it's quite easy to see if you do like linear regression, for example, that delta gamma, the regression coefficient is just going to be this times this averaged over. So this correlation vanishes. So you get a negative term because x0 is -1. And so ignoring a bunch of details, so you're going to get a constant and you're going to get 2 times the average of psi square over the backgrounds, the 150 backgrounds plus some noise. And this noise is Gaussian distributed. Okay, so that's all we are saying, right? So now you have this regression coefficient that you get, and that's always going to be positive, which means that the slope is always going to be negative, and it's going to be linear with Gaussian fluctuations. So that pretty much solves the problem. And so this quantity turns out to be quite an interesting quantity to think about, right? So if you just look at this quantity, this is the expectation of psi square over variations in the background. So you're flipping your xi's over plus or minus 1. And so we're going to call this V0. Because this V0, if you write it out in terms of the coefficients, it essentially captures how much that particular locus interacts with everything else related to the variation that you get from everything else. Right, so this is, again, like I was mentioning, this is a Fourier transform. So if you take square of the expectation, you're just taking the sum of the squares of the individual coefficients. You can take dot products and so on. So it's really easy to kind of write this out. But effectively, your V0 ends up being this. And this is a global measure of how much this locus couples to everything else. Right? So this kind of assay gives you that, which you cannot get in other kinds of assays. And this turns out to be quite an interesting quantity. This quantity is always positive or non-negative, right? The other feature of this is that in this limit, the variance of these fluctuations are going to be proportional to the slope. Right? So the variance of the noise will be proportional to V0. That's something that we can test in the experimental data, and we did, and it turns out to be true, right? So it kind of validates this mechanism. The theory is quite simple, but I think it tells us a lot, right? So it tells us that it's useful to think about many dots over here, that we should also think about many dots over here. So each locus interacts with many others. This one in some senses is quite a strong statement, right? So this is the correlation between these two. And if you write it out, this is effectively saying, so let's just focus on the first order term. This is basically saying this, the sum of products of the additive and the epistatic coefficients is much smaller than the variance. So that's going to be something like this, right? So that's the comparison that we have to make. If we ignore all the higher order terms. So we're, sorry, this has to be Fi0. So we are saying somehow that you have the sum of the additive effects and the epistatic effects. The additive effects are typically larger than the epistatic effects, we think. But these kind of average out, right? So these scale as square root of L because they can have any sign. They can have plus, positive or negative sign. So if you assume that these are essentially like in principle, Fi, let's say is normally distributed with variance sigma 1 square. Fi zeros have some other scale that's defined by the variance. This one's going to scale as sigma 1, sigma 2 times square root of L. Whereas this one's going to scale as sigma 2 square times L. Yeah.
This is because of the way the experiment is set up, that there's no demography and there's no evolutionary history. That is just because you cross two unrelated strains.
Yeah.
And how much of this is like reflects the particular experiment, and how much do you think it reflects like in real life where you have demography and history that will cause correlations for sure between these coefficients?
Yeah, I think it's a good question. I think one of the key challenges I would say is that everything that you do is dependent on how you set up the experiment and the context. The hope or the belief is that, or the philosophy is that you do this enough times over different systems and you get some general picture. And I kind of want to reiterate that we don't want to infer these specific values, but we want to say something general about this. Yeah.
So again, I'm going to still be naive to the non-biologist in the room, but if I understand correctly, so a question you have is like, how sparse are these Fs? Like, are a lot of them very close to zero, or is it a pretty non-sparse expansion that has like lots of interactions that are? So
yeah.
So are you saying that the fact that your experiments conform with a simple model suggests that these expansions are not that sparse and a lot of the Fs are not?
Yeah. There.
Or do you somehow know that a priori? That's what I'm trying to understand. Like, what do you expect a priori from theory? And
Yeah.
or are you saying you're learning about the Fs from these?
So a priori, we don't know, right? So we couldn't measure. For a long time, we couldn't make the kinds of precise measurements to infer the Fs. And we still cannot in many cases. So we have to come up with an indirect way. And this is suggestive, though not conclusive, that you should care about the higher order terms, but it doesn't tell you how many or their magnitude. All it tells you is that there's enough that you can use a concentration argument. That's all it tells you. Yeah. And I think in general, like we're interested in the statistical structure of these Fs, right? So we want to kind of come up with a statistical description of these Fs. And I'll get to that just like in a few minutes. Yeah.
Is it fair to say you had to work with this experimental setup to get a situation where you're averaging over many genotypes?
Yes.
And then you see this simplicity.
Yeah, I think before these kinds of experiments were done, you're often working with very few loci. This is kind of, there's a series of technologies that allow this to be happen, right? So like first of them is sequencing. The other is the fact that these are liquid handling robots doing the experiments. Which makes a huge difference, right? So and there's all this DNA barcodes that you need to use to keep track of so many different cells at the same time and measure their fitness. And I think Michael really kind of pioneered many of these techniques. Yeah. All right. Okay. So I'll say, so we got to point number 3. I want to say one more point, which is that you can measure this from experiments, right? So you can do this for the 100 mutations in your experiment. and you can get a distribution of these. And if you look at it, it turns out to be about, it looks somewhat like an exponential. I don't know what to make of it. That's what you get from the data, right? So it's always going to be positive, and that's what you see, right?
It's the horizontal axis.
It's V, and this is P of V.
But the numbers I can't.
0.15 and 0.3. I don't know why I picked, didn't round them up, but yeah, that's all right. Feel like I wanted to make one more point. There was point number 5 in my notes, but I forget, but I don't think it's that important if I forgot it. All right, so what I'm going to do now is to go into theory land for the next, I guess, 15 minutes that I have, or maybe 10 minutes because I have 5 minutes for questions. And I wanted to write down a very kind of phenomenology-driven conceptual model of how evolution could play out in a landscape that satisfies these various constraints that we got from the data. The constraints are very weak. Yeah.
Probably when they saw that you checked the noise thing empirically in the data.
Yeah. So we checked that the noise is proportional to V0, and it does actually work, right? Right. So I'm going to write down what folks call as a fitness landscape model. It kind of echoes some of the work done in statistical physics, but it's slightly different in spirit from what one does. Okay. So the way we define ensembles is slightly different than what folks normally do, and I'll explain how. So let's say we get back to our, I'm going to ignore the constant term in this expansion because it's not important. And as I mentioned, we want to say something about the statistical structure of these Fs. I want to construct a model that satisfies this constraint, this one. This is something that we want to put into the model. And the final point, which is the most important one, which I actually forgot, is we want a model where the distribution of fitness effects is a function of just the fitness. This is quite non-trivial, right? So that's something that we want to kind of come up with a model that has all of these properties, right? So, and the way we're going to define the model is to say how these Fs are generated. And as these models go, the standard way of defining these models is to say that, yes.
We're going there. Didn't you have an intermediate class that were threshold dependent that wouldn't fit into that equation?
Yeah, that's right. So there's definitely mutations that don't fit into this picture.
But just for those, you're going to talk.
Yeah, we're going to focus just on the ones that do have interactions because they're the most interesting ones. All right. So the way you define these models is to specify a way to generate these coefficients. This kind of echoes many of the spin glass models in statistical physics, right? So you kind of specify what these variances are, and you assume they're all the Fs are independent, they're all Gaussian, but the variances can be different. So the way we're going to define our model is to say that our sigma I square is going to be some locus dependent factor mu I. So that's specific to a locus. Sigma ij square is going to factorize this way. And sigma ijk square is going to factorize this way, and so on. It's a very particular structure. The effect sizes of each of these terms goes down with order, but the number of those terms goes up. So they do contribute, right? But that's the structure that we're going to impose. And we're going to say these mu i's. So L is much larger than 1, as things typically go. We're going to define a distribution over mu. And we're going to assume that the average value of mu is much smaller than 1, which kind of echoes that. It's about 0.15. And we're also going to assume that mu bar L is much larger than 1. So you need to get this, you need to assume this so that enough of the larger higher order terms matter, right? This is what we call the epistatic limit. This is the interaction limit. Okay, so this is the model. As these things go, what you get out of this model is first it's a way of sampling your landscape. So you sample your Fs, you freeze them, right? So now you've frozen your Fs. So you sample Fs from this generative process. And the way we're going to perform our numerical experiment is to sample the genotype that has a particular initial fitness. So you sample many of them and you condition on gamma 0, and you apply and you perform a numerical selection experiment, and you're going to see a sequence of fitness changes and mutations that appear in the sample. And now you can ask, what is the distribution of next the fitness changes that you get, given that you've seen the sequence. Now, the advantage of working with these models is that everything becomes a Gaussian process, right? So if you average over the ensemble of X's that you initially sampled from, given fixed Fs, these variables are jointly Gaussian, which means that the conditional distribution of the fitness effect is also Gaussian, which means that if the conditional distribution is Gaussian, it means that you can use linear response theory, right? So you can write your fitness effect as a sum of plus some noise, right? So that's something that you can generally do for these kinds of Gaussian models. Now, these coefficients over here, or Green's functions as we like to call them, they depend on these Fs in some complicated way. But the important point is that they average over many Fs. So they're usually the sums of many Fs and so on. So we're going to assume that these Ks actually self-average. So we can replace this case with the ensemble average that we're drawing these Fs from, right? That's a strong assumption. And it turns out that for this particular landscape, all the Ks for K greater than 1 are 0, are identically 0. This is a special property for this particular landscape. It's memoryless, right? So this is basically saying that delta gamma, and you can show that the first one, which is K0, is minus 2V, and it turns out that in this case, it's time. V is mu over 1 plus mu, but it doesn't really matter. But ultimately, what you get is that in this particular landscape, this is true. So the fitness effects that you're sampling from depend only on your current fitness, and there's Gaussian fluctuations on top of that. And your mu is sampled from some P of mu. And this is a selection. This is an evolution experiment. So you're sampling only positive fitness effects. So the probability that you select a mutation, if you assume the simplest possible selection procedure, which is called strong selection weak mutation, if you're familiar with it, is to say that P fixation is 2 times delta gamma if it is positive. If it is negative, it's 0. If delta gamma is positive, then you select that mutation in proportion to that. And these terms are always small, so this is not going to go more than 1. Okay, so that's really the process. So you can do this. You can run this process in your computer pretty easily. You can get predictions for what the fitness trajectories will look like, and it should be pretty clear that this is going to produce very reproducible trajectories, even though the specific sites that you mutate are going to be very different. There's one particular thing I want to mention. Maybe I'll mention it in words, even though I was going to write down some equations, is that this is like a ratchet, right? So this is like you have some regression to the mean, but you're selecting only the positive fluctuations. So initially this term is small. So you're selecting fluctuations and all the positive ones get selected because of this. But eventually this becomes large enough. And at some point you're kind of being pulled back, and these are particularly large deviations in the fluctuations that get selected. So there's a large deviation argument that you can use to calculate what is the distribution of delta gamma for very large values of gamma. And it turns out to be an exponential generically. Like there's a large deviation argument which you can show, you can write down, which shows that the probability of a positive fluctuation, this ratio basically becomes something like this. And in this limit, when gamma is large, you're always selecting mutations that are in some sense intermediate. Like you don't select the smallest, the mutations that have the smallest effect because they will have zero effect. You don't select the ones that are too large because they will have a pretty large, they're going to break everything. You want to select the intermediate ones, but the effect size of those intermediate ones decreases as you go up the landscape. So if you run this process, you can show that you're going to go like this and then keep going. You have a linear phase over here. This is gamma as a function of time in your simulation. There's a linear phase over here, and then you have slow increase, which you can show is I mean, for this particular, it doesn't matter. What the exponent doesn't matter, but it's a t to the 1/5, right? So you get a gradual rise in the fitness. So this is a special property of this model. You don't get this for all models. If you take a classic ratchet, where mu is fixed, the viscosity is fixed, for example, this is going to be a Gaussian, the DFE is going to be a Gaussian, right? So this is a Gaussian distribution, mu is fixed, so there's a Gaussian that is moving to the left as your fitness increases. So you're not really going to go beyond a certain value. Typically, if your mu is fixed, you're going to get something like this. It goes to a particular value and then flattens out. So it does matter that you have mutations that have a large range of interacting coefficients. Yep. All right, so that's the theory part of the, yeah.
So there's this other model, right, that you run out of mutations, that there you would expect from extreme value that it's square root of log t, right? And so can you do an experiment to distinguish that?
A numerical experiment?
No, I'm just wondering, like, can you measure this? Because I mean, that's the other thing people say in the community, right? It's just you're running out of mutations. So then you're sampling a gumball, so then you'd get expected to be square root of log t. I'm just wondering if you thought about whether you have the resolution to tell t to the 1/5 from.
Yeah, I mean, there's a lot of assumptions going into this model, so I wouldn't even do this experimentally. I really view this as a conceptual model.
Right, the other one is that you just run out of mutations, right? Which is what most people do.
I think there's many mutations that you can potentially do in a real system, right? So like when these models were run a long time ago, they were working with first they're working with just models. They're not working with experiments. And in the models, they always assumed that, you know, you had a finite set of mutations and you ran out. I just want to say one closing comment, which is that the way I think trying to get back at the original question about predictability and repeatability, there's a lot of pathways in the cell, right? So if you think about what's happening inside the cell, there's many, many metabolic pathways. If you've seen these pictures of a cell, like they're extremely complicated. So one can ask like why, you know, this picture suggests that everything is somehow clustered in one pathway, right? So really the central hypothesis here is that all the perturbations that you do are somehow strongly coupled. So they're all interacting in one pathway. So for whatever reason, everything seems to be coming from here and they're all talking to each other. So what's happening here, right? The way at least we've been thinking about it, at least I've been thinking about this, is that there's many pathways. All of them are hyper-optimized. You know, the glycolysis pathway is hyper-optimized to metabolize glucose and so on. But there's only certain knobs that you tune when the environmental fluctuations change, right? So how you allocate resources to different pathways changes as the statistics of the environment changes. And there's really, even though there's many things that matter to fitness, there's only a few flexible knobs. And all the mutations that we are interested in cluster in those knobs, and they interact because they're all clustered in those few knobs. So the broader goal right now is to figure out how many knobs there are, whether this picture is actually true, and ask from a more systems biology perspective or conceptual perspective, why are there so few knobs, right? So I think that's really where this is headed, in my opinion. All right, thank you.